Okay, today's video is all about your nervous system. So how do you respond to stimuli, whether it's internal stimuli or external stimuli? There are two systems, your central nervous system and your peripheral. Your central nervous system is your brain and your spinal cord. Your control, it works as your control center. So it is receiving and interpreting stimuli and sending out responses. So in class, we're going to be dissecting a brain, looking through, finding different parts of the cerebrum, the pons, the hypothalamus, pituitary gland, medulla, cerebellum, all those kinds of neat things. All right, peripheral nervous system. This is what extends out from your central nervous system. So all the nerves that extend out from your spinal cord and from your brain to the rest of your body. They are divided up as follows. I thought this was the easiest way of doing it, so I'm going to uncover it all first. I made it into a little flow chart. So you have sensory neurons and you have motor neurons. Your sensory are all the ones that receive the message and send it to your central nervous system. Your motor are all the ones that go out. They are divided up into somatic and autonomic. So somatic control voluntary muscles. So all of your, like if you want to do a bicep curl, that is a somatic nerve going from your spine out to your bicep and causing it to pull that muscle in. You are thinking about it. Autonomic, think automatic. It's doing it all on its own. So that is involuntary muscles. These are divided into two categories, the sympathetic and parasympathetic. When you see the S for sympathetic, I want you to think speed up. If your body has to respond to something that's sort of like an emergency situation, you do what's called fight or flight response. When you need to respond to something, you're going to need energy. So you need your heart rate to go up. You need oxygen, so you need your breathing rate to go up. Don't worry about digestion. Digestion is like turned off at this point. Instead, take any available stored glycogen and convert it to glucose. Get your body geared up for action. The other is more what we think is like active during your calm time. This increases digestion. So as long as I'm not busy doing everything else, I might as well digest my food now. Reduces heart rate and breathing rate and calms your body down. So I have this diagram, which I'm going to print for you for class, and it goes through, shows you different regions of the brain. I have another one too that shows where the pituitary and hypothalamus and all those things are. And then if you go down, so that was all central nervous system. And then as you go down, it's broken into the peripheral nervous system, both autonomic and somatic. So somatic is all going to muscles that you control. Autonomic is what is involuntary. So some of that is parasympathetic, rest and digest. And then sympathetic, fight or flight. All right, now another one, I'll print this one for you as well, probably cover up some of the things and have you think about what's going on in each step. All right, nerve cells. So now when we get into a little bit more detail, what does a nerve cell look like? The structure, I want you to draw this. This is the cell body. The extensions out are called dendrites, and receptors are found on the dendrites. And you can find several different types of receptors because there are different chemicals in your body that would be received by a nerve cell. There's a nucleus in the cell body region. There is a long stretch called the axon. When we do the cat dissection, it's really cool because you're going to be able to see neurons that are really long. And in your leg, you can think about there are neurons that run along the bone, so they are also very long. So that long stretch is called the axon. It can be insulated with what's called a myelin sheath, and it might have these little gaps, places where it comes in. It allows saltatory conduction, which means it can actually conduct impulses even quicker. These would be called the nodes of Ranvier, where it comes in a little bit here and here. The end is called the axon terminal, and the space in between is called the synapse. All right, there are different types of neurons. The first is sensory. So sensory, receive the stimulus from your sensory organs, pass the message to your central nervous system. So a great example, your optic nerve. When we dissect the sheep eyes, you're going to see the optic nerve going from the back of the retina to extending out to the brain. Your interneurons are in the central nervous system. They are what are in your brain and your spinal cord, actually receiving the impulse from the sensory neurons, interpreting it, and sending out another impulse to the motor neuron. So interthink, those are the connections, they're in the middle. Motor neuron, the last one, these extend from your central nervous system, so either from your brain or from your spinal cord, out to effectors. Effectors are what are actually going to do the work. Those are your muscles and your glands. They receive impulse from the interneuron and they cause a change. So they're going to either stimulate a gland to release a hormone or they're going to stimulate a muscle to contract. All right, a reflex. I know you've all learned what a reflex was before. It is the simplest pathway. It never goes to your brain, so you do not think about it. It is innate, and it's usually very protective behavior. If someone was sitting up here on a chair and I took a hammer and banged their knee and their knee flexed out like this, that is a natural reactive reflex. So they didn't think about it, their knee just jerked. Or if I punch like this and someone blinks, same type of behavior. The pathway is it goes from sensory to interneuron to motor neuron. Here's the difference though. The interneuron has to be just a neuron in the spinal cord. Here's what one looks like. So here is the sensory organ. You can see the skin right here. You can think of the example of touching a hot stove. I think everyone thinks of that when they think reflex as well. You touch a hot stove and right away you pull off without even thinking about it. What happened? The receptors in the skin sent a message through a sensory neuron to the spinal cord. The spinal cord interpreted the message and quickly sent a message out to the muscle to move your hand away. All right, a nerve impulse. These are the steps of what do you actually have to do to send a message. So it is an electrochemical message sent by nerve cells. Part of it is electrical because it changes the polarity of the cell. So the polarity is the charge. The charge is going to change. The other part is chemical. The chemical messages are called neurotransmitters, and that is what's needed to fill the gaps. There are spaces between nerve cells called synapse, and this is what connects nerve cells. All right, action potential is another word for a nerve impulse. Neuron at rest. So before we talk about what it's like to have an impulse, you have to talk about what does the neuron look like, what's its charge before the start. So there is a difference in ions. There are more Na plus outside of the neuron than in. And there are more K plus, potassium, inside the neuron than out. This leads to an overall negative charge inside of the neuron. This gradient and also negative DNA inside the cell explains the polarity. So polarity means a difference in charge. If something is polar, you can remember that from chemistry, that means one is more electronegative, it's pulling more than the other. So anytime you see polarity, that means there's a positive and there's a negative. The negative here is inside the neuron and the positive is outside the neuron. So negative 70 millivolts is a pretty normal resting potential. Could be anywhere between negative 0 to negative 100. Anywhere there it could be. Polarity is maintained. So how do they keep the Na plus out and the K plus in? They're pumped. So the sodium-potassium pump keeps the ions pumped out to the other side, and it keeps them against their gradient. All right, here's a picture of what it looks like at rest. So here's the lipid bilayer of the axon. Here's the inside of the neuron. Here's the outside of the neuron. All of these little purple dots are sodium, and it says here the sodium-potassium outside is 450 millimolars. And the potassium is only 20 millimolars. But inside, the potassium is 400 and the sodium is only 50. So that difference in charge is maintained by this sodium-potassium pump, uses ATP and keeps pushing it against the gradient. Sodium out from low to high, potassium in from low to high. So this one has a membrane potential of negative 60 millivolts. So its polarity, its difference in charge, is negative 60 when it's at rest. All right, if we want to have an action potential, we want to forget the rest and actually send a message. No more resting, but instead have something happen. So here are the steps. First of all, there has to be a receptor. There has to be a ligand that is received by the receptor, and it's got to cause ion channels to open. So sodium gates can open, potassium gates can open. Some cells even have chlorine gates. So there has to be something that's opening and closing. Now, there's two different types of postsynaptic potential. So postsynapse, if you think about this for a second, if my hand represents a neuron, here are the dendrites, and here's the synapse, the space. Well, at the end of one neuron that came before it was the axon terminals. This would have to let out a neurotransmitter that would get picked up by this next neuron. So this neuron is postsynaptic. It's after the synapse. It could be an excitatory postsynaptic. Excitatory means, oh, let's open up the gates, let's have a change happen, and let's have it be positive. Let's depolarize. So an excitatory postsynaptic potential opens up gates and depolarizes the membrane. Now, there's another new word here. It's called threshold. A threshold is a certain minimum number that the depolarization has to get to in order to open the rest of the channels. The other type of postsynaptic potential is inhibitory. Gates still open, but it actually opens for ions to move the opposite of what we expect, and they actually further polarize the membrane, making it way harder to reach the threshold level. So instead of, here we are at negative 60, instead of going up like this and depolarizing the membrane, it actually takes it more negative, makes it less likely to send a message. So the first step is to have the receptors receive this message, and it's going to have to be excitatory if it's going to send a message. If it's inhibitory, it makes it not work. So let me just give you an example of this. In your brain, you have some neurotransmitters that are inhibitory, meaning they do not want you to feel pain or feel something over and over and over again. So you know if you slam your finger on something, it hurts really bad, but then after a while you're like, oh, I sort of forgot about it. I don't feel it anymore. Does it still hurt? Sure, but you're not feeling the pain because your brain has said, you know what, stop sending the message. They know that the pain is there. So that would be an inhibitory example. There are some neurotransmitters in your brain like GABA, G-A-B-A, that work to inhibit certain responses. All right, step 2. If the threshold is met, all sodium-gated channels open along the axon and sodium rushes in. That's going to cause depolarization. Instead of being at negative 50 or negative 60 or negative 70, it opened the gates, it let the Na plus in. The number now is rising up, maybe closer to 0. This is also known as a wave of depolarization. All right, next step. The potassium gates open. They see the sodium gates open. Well, they don't see anything for real like seeing, but once one opens, it tends to open all of them. So sodium gates are open, so potassium is like, well, I guess we'll open too. So they open and potassium starts moving out. That process is called a repolarization, because if potassium goes out, now we're taking the positive out of it and we're going to bring that number back down. I'm going to do a little graph with you at the end to make sure all these numbers make sense. All right, next step. Sodium-gated channels close quickly and potassium-gated channels close slowly. So once these have all opened up, they say, well, shut the door. When they shut the door, sodium gates close very quick and potassium takes much longer. Sometimes that causes this to happen, hyperpolarization. So if the sodium closed quickly and the potassium closes slower, more potassium might go out than you actually wanted to get the membrane back to polarize. So instead of going back down to negative 60, it might drop down to like negative 80. All right, next step. At the end of the axon, where the terminal is, right before the synapse, a message has to be sent to span the gap. That message is called a neurotransmitter. It is a chemical. In order for the chemical to get out, calcium has to come in, bind to one of those exocytosis vesicles, and let the contents out into the synapse. So calcium binds to vesicles containing the neurotransmitters and causes exocytosis. The neurotransmitter is released into the synapse. All right, recovery. Basically, if you think about it at the end, everybody's on the wrong side. Sodium is inside, potassium is outside, and they got a flip-flop. So who works to do this? That sodium-potassium pump works to get everything back to the resting potential. No new impulses can be sent during this time because the ions are not in the right places and the resting potential is not back to normal. All right, continuation of an impulse. In order for that impulse that went through one neuron to go to the next neuron, we need neurotransmitters to be released into the synapse and start another impulse by binding to the receptors on the next neuron's dendrites. That has to happen to continue the signal. So if I make a quick diagram, here's one neuron. Here's a receptor. It picks up a ligand and it opens channels. When it opens the channels, the Na plus moves in. This is now going to change. Let's say it was -60. It is now depolarizing, which means it might go up all the way to a value of 0. Potassium responds and K plus gates open and K plus moves out. That's going to repolarize our membrane back down to -60 or maybe even lower. At the end, there are calcium gated channels. Calcium comes in, binds to these vesicles, lets these neurotransmitters out into the synapse. Now, the next neuron can pick up these messages, and the same thing can happen again, over and over and over. All right, lastly, I want to show you a diagram of an action potential. Here is my normal resting membrane potential. Normal, 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 normal. Starts going up. It only goes up if the Na plus gates open. If it reaches a certain minimum amount, this is called my threshold. If it meets that minimum amount, we get the spike. The spike